In this video, we will discuss about the M phase, that is mitotic phase after the interphase. In this mitotic phase, let us define it first, okay? So it is nothing but the division of the parent cell, okay? The parent cell will first grow and it will divide. After dividing, what happens is that the two daughter cells are formed. These two daughter cells have the same similarities of that of their parent cell and they have the same number of chromosomes. This all happens in the M phase, that is mitotic phase. What is the significance of mitosis? Mitosis will happen in order to restore the nucleo as well as cytoplasmic ratio. Whatever ratio will be present in a cell, what nucleus should occupy, how much the cytoplasm should occupy is all fixed. So to restore that, the cell division plays a very important role. And the second thing is that to replace the dead or worn out cells, whatever dead or worn out cells are there, it can be replaced by the process of mitosis. And second, in order to increase the number of cells inside an organism, mitosis plays a very important role. So this was about the significance of the mitosis. Then mitosis happens in four phases, as we know: prophase, metaphase, anaphase and telophase. So let us study each of them in the detail with the diagrams so that you can understand the concept easily. So let's start.
So first, moving on to the prophase. We know that in prophase it is the longest phase of division. In case of the mitotic phase, the prophase is the longest phase here, and it is shortened and thickened of the chromosomes. What happens here? The shortening as well as thickening of the chromosomes. In the diagram you can see the chromosomes, okay? There it will shorten as well as thicken itself. Okay, the chromosomes whatever are there, no, for they will split into two chromatids, the single threads. Okay, it will split into two chromatids, and those chromatids are held together by a centromere, that is nothing, or you can say it as a kinetochore. Okay, and these centrioles, they will develop very fine eyelashes like threads. They are called as astral rays. Okay, these centrioles along with the astral rays, they will form something called as asters. Okay, and the two asters will start moving towards the opposite poles. By the end of this prophase, they will reach the opposite poles. Okay, you can see those things in the diagram while I'm explaining. Next is between the two asters, there will be development of a very fine proteinaceous thread. Those threads are called as spindle fibers. I will show you in the next picture. You can see the spindle fibers in that, and these asters along with the spindle fibers, they are called as spindle or mitotic apparatus. Such as spindles is called amphiastral spindles. Okay, and the chromosomes will get attached with the spindle fibers from their centromeres. Okay, this you can see in this phase, and the nuclear membrane and the nucleus will start disappearing slowly in this phase. In this next above the late prophase picture, you can see that how the nuclear membrane has been disappearing slowly and the spindle fibers are been formed here. In the metaphase, mainly the chromosomes will be arranging themselves between the poles and the equator of the spindle, and this arrangement will form a plate. We call it as equatorial plate, or we can say it as a metaphase plate. Okay, and the spindle fiber, or called as microtubules, get attached to the kinetochore or the chromosomes. Okay, I will show you the picture of metaphase. See how there is arrangement of those chromosomes in the equatorial plate, and the metamorphic plate also you can see there, and spindle fibers are formed. This is what the metaphase.